At this year's ALKS 2025 First Time Disclosures Symposium, we saw a range of groundbreaking clinical candidates, each leveraging innovative chemistry to tackle historically undruggable targets. Below, we analyze the market potential and scientific differentiation of 4 highlighted candidates. Number 1, IAMA6 from Iama Therapeutics. This selective NKCC1 inhibitor is engineered to avoid off-target NKCC2 activity while still crossing the blood-brain barrier, or BBB. Given the high unmet need in CNS disorders and the historically poor druggability of ion channels, particularly those linked to fluid balance, we estimate peak sales of $1.50 to be plus if clinical safety mirrors preclinical selectivity. Avoiding diuretic effects, which are linked to NKCC2 inhibition, also positions IAMA-6 as a first-in-class neurologic agent. Number 2, BP1664 from Repare Therapeutics. This ADME-optimized PLK4 inhibitor features a cycloprobal substitution that enhances potency and selectivity versus earlier PLK4 inhibitors. With several ongoing trials in TRIM37-amplified cancers like triple-negative breast cancer, BP1664 could offer a differentiated profile versus existing cell cycle inhibitors. If it shows efficacy and reduces the toxicity observed with earlier candidates, we see peak sales of $800 million minus 1 to be in these niche indications with potential expansion to broader solid tumors. The cycloprobal substitution exemplifies how structure-based design can overcome limitations of previous scaffolds, enabling more effective kinase inhibition. No. 3. ALKS 2680 from Alkermes. This C-shaped OX2R agonist is stabilized by a covalent linker that keeps the molecule locked in its bioactive conformation. As orexin signaling modulators, ALKS 2680 targets the same pathway as the small molecule antagonists like suvorexant, but with potentially improved binding kinetics. If it demonstrates superior efficacy and safety in narcolepsy, idiopathic hypersomnia, and circadian rhythm disorders, we see peak sales of $2 billion as a result. Notably, ALKS 2680 represents a new approach to GPC or agonist drug discovery via conformational stabilization with covalent tethering. And note 4, BMS986-38 from Bristol Myers Squibb. This macrocyclic PDL-1 inhibitor features a fatty acid PEG modification that enables extended half-life through albumin binding. This next-gen immuno-oncology agent could enable weekly or oral dosing, potentially improving compliance and expanding the use of checkpoint inhibitors into the adjuvant setting. Given the durability of anti-PD-1 and anti-PDL-1 therapies in various tumor types, even those resistant to current checkpoint therapies, we believe BMS986-38 has blockbuster potential of more than $1 billion in sales. By merging the stability of a macrocycle with the advantages of half-life extension, BMS is bridging the gap between antibodies and small molecules in immuno-oncology. In addition to these 4 candidates, ALKS 2025 showcased a range of other exciting science, including metalloenzymes, chromatin remodelers, BBB-penetrant kinases, and more. These breakthroughs highlight both the power and the challenge of modern medicinal chemistry. Overcoming these challenges will require innovative solutions in areas such as retrosynthesis and scale-up planning. Thanks for watching.